Thank you all for attending MP Biomedicals' The Path of Discovery talk at Experimental Biology 2021. As many of you know, animal models are such a crucial part of research. They help us to understand the basic mechanisms of human physiology to aiding in the discovery of therapeutics. In fact, animal models are still the gold standard for studying the mechanism of action for diseases such as colitis and cancer. At MP Bio, we raised the standard of in vivo research by maintaining the health of your animal model, as well as to provide the appropriate reagents for your downstream assays to determine mechanisms of disease action. With our standard and customized animal diets to our anti-tumor agents and pharmaceutical-grade APIs. Animal models can be so sensitive and require special conditions, treatments, and specially formulated diets for the maintenance of health, but also custom diets for the animal model of interest, such as high-fat diet studies, including obesity mice or colitis models with specially formulated fatty acids. Whatever your needs are to maintain healthy animals or answer specific questions, MP Bio offers the full spectrum of these options to fulfill your research needs. Looking to mimic disease in your animal model? MP Bio's products can help with your complete mouse model of human disease workflow. Let's take, for example, colitis research. Chemical induction of colitis is the most widely used method. MP Biomedicals offers DSS, dextran sulfate sodium, cited in over 10,000 publications, and is the gold standard in colitis research. And now, we are excited to be the first to provide you with the average molecular weight of DSS to further help you fine-tune your experiments, save time, and money. So now that you've established your animal model, the real work begins with your experiments. You want to verify your animal model by collecting tissue and analyzing tissue samples, and examining the cell morphology and histological pathology to better understand the disease condition. MP Bio offers reagents and dyes such as a simple hematoxylin and eosin or H&E stain for histological pathology scoring, to more complicated and specific immunostaining antibodies for protein analysis to meet your animal model verification needs. Need to verify your model at a molecular level? We can help. Our fast prep homogenizer and large selection of lysing matrix provides you with a simple yet versatile solution to help you isolate nucleic acids and proteins, whether your sample is fresh or preserved, hard or soft, tough or fibrous, and this is not limited to animal tissue. The fast prep and lysing matrices are ideal for any type of sample you would want to homogenize to retrieve nucleic acids or proteins. Now that you've homogenized your sample with the fast prep and lysing matrix, we offer a variety of nucleic acid and protein extraction kits, such as our Fast DNA Spin kits for feces and animal tissues, Fast RNA ProGreen kit and Rapid Pure RNA tissue kit, and the Fast Glycoprotein isolation kits. And these will make your downstream analysis simple and efficient. Once you've processed your samples, you're ready to analyze your sample using downstream applications. We offer a collection of polymerases such as MasterMix, probe kits, PCR, qPCR, as well as PCR product cleanup with our Gene Clean PCR purification kits that allows you to quickly analyze your DNA and RNA. We also offer products to help you visualize your nucleic acids and proteins with MP Bio's electrophoresis products, such as agarose powders or precast agarose gels, Western blotting with our antibodies and acrylamide, and immunostaining through the use of our FTSE conjugated antibodies against a number of proteins, including but not limited to housekeeping genes and disease marker indicators such as complement C3 for your colitis model. Though animal models are highly valuable, they can be quite expensive. MP Bio can help you maximize the use of your animal model by also offering cell extraction reagents to help you examine at a cellular level disease pathology, such as our in-house validated lymphocyte separation media with high yield and greater than 95% lymphocyte cell viability. Now that you've validated your animal model, you may want to go back to the in vitro cell culture model to examine more detailed cellular mechanisms. Our cell culture reagents portfolio is designed to meet your needs with media, both liquid and powder, and the necessary components for healthy growth, such as antibiotics, serum, whether it's bovine, human, or from another animal source, growth factors, and supplements to contamination surveillance with our mycoplasma detection and removal reagents. So, ready to treat your in vitro model? MP Bio can help with our anti-cancer reagents, as well as our anti-inflammation reagents to meet your research needs. So what if you're not doing colitis research? Well, MP Bio is not limited to colitis research. In fact, we can take the same approach and apply it to any type of animal model you have, whether it's the spectrum of cancers, the many disease models, or stress research. MP Bio can help you on your path to discovery.